Okay guys, so I'm going to go through this nice and quickly. Number one, what are the three differences between RNA and DNA? So the first is what sugar they have. So DNA has deoxyribose while RNA has just ribose. The second thing is while DNA is double-stranded, RNA is single-stranded. And finally, there's one base difference. It's that RNA has U while DNA has T. Three different types of RNA. Here they are. You've got mRNA, tRNA, and rRNA. So for things for you to keep clear in your head, mRNA is the long strand that has 3 nucleotides which make up a codon. Okay, they carry the message, but specifically they're what are going to tell the tRNA what amino acid to bring. So the tRNA actually brings the amino acid to the ribosome, and the way that mRNA tells tRNA what amino acid to bring is that tRNA has an anticodon. Now the anticodon is complementary to the codon, right? So if, for example, this is ACC, this would be UGG. Okay. For rRNA just makes up the ribosomes. Okay. For transcription occurs in the nucleus, and what happens is DNA is copied into or made into RNA, and what does this is the RNA polymerase. Okay. Before the RNA can be used to make proteins, we first have to edit it so the introns are removed while the exons remain where they are. For translation occurs in the cytoplasm. What's happening in translation is RNA is being used to make proteins. Okay. Specifically, once you have a long line of amino acids, they're connected by a peptide bond. Okay. And that peptide bond creates this long string, which we call either a peptide chain or a polypeptide chain. And once you then take that long chain and kind of twist and turn it to make a 3D structure, that's when you get proteins. Okay. In order to make a protein, a group of 3 RNA nucleotides are read at a time. These are called the codons. Now, tRNA brings the amino acids to the ribosome where the peptide chain is made. The anticodon, so the anticodon is from the tRNA. The anticodon matches the codon from the mRNA to dictate what amino acid is added. So I already just talked about that. All right, we got point mutations, we got chromosomal. So here this is substitution because instead of adding in something, I've completely gotten rid of my G and substituted a Q for it. Here for insertion, I've inserted a Q. Here I've deleted my G. And these are called frameshift mutations because what happens is it messes up the way that I'm reading. So rather than staying A, B, C, then D, E, F, then G, H, I here, I've shifted the reading frame because I go 3 times at a time. So all the amino acids after that mutation, they're going to be messed up. Okay, chromosomal, here I deleted it. Here I duplicated the B, C, D. Here I inverted it, so I flipped it. Right? So instead of B, C, D in that order, it's backwards. And here I translocated a section. So here I've got this rando section that has no place, and so it's translocation. Remember, chromosomal is big chunks of chromosomes. Point is just a single change in the nucleotide. Okay? Most mutations are C, they have no effect. Beneficial means they're helpful. Detrimental means they hurt. C, have no effect. So most have no effect. In prokaryotic gene regulation, what's happening is we're using operons. And so what will happen is a repressor will be put onto the operator, and that prevents the copying of DNA into RNA. And so specifically, it prevents RNA polymerase from copying DNA. Okay? Now, this example with lactose, what happens is the repressor, what would go onto the operator to stop the RNA polymerase from copying, that is not present because, and the reason why this makes sense, what's being made, what's being transcribed and then translated is an enzyme that will break down lactose. So the only reason why I would want this translation and transcription to be happening is if lactose is present. And so that's why it's turned off, meaning why the lactose enzyme is allowed to be made, because lactose is present. The other example was with tryptophan, I believe. And with that, if tryptophan is present, then this transcription is turned off. And the reason why that makes sense, what's being made is tryptophan. So if I already have plenty of tryptophan, why make more? And so that's what happens. When tryptophan is present, it's turned off. So it's the exact opposite of the lac operon. Okay. Now, this is all for prokaryotes, the concept of operons and operators and everything. For eukaryotes, we really didn't get into this in a lot of detail, but we use transcription factors. The important thing for you to know with prokaryotes and eukaryotes is the fact that it's RNA polymerase that is transcribing the DNA. Okay? 9, the previous forms of gene regulation prevent RNA from being created in the first place because it's preventing transcription from happening. Another way to prevent a protein from being created, we call RNA, I wrote splicing, it's silencing actually. Okay, I believe, either splicing or silencing, I'm sorry, I can't remember for sure. Okay, in this process, what's happening, we've got microRNA, and they form with other proteins, they form a silencing complex, and they literally chop up mRNA. So the way that this is different from these guys, in these guys, this is preventing transcription from happening. Here what happens, transcription has already happened, now it's just going to chop up the mRNA that was formed. Okay, so if you look at 10A, here I've transcribed, so these are complementary to that. Here I then looked at my chart and got this, so I don't think you need me to explain it, I'm hoping. There are 64 different combinations, and you guys can literally count it or just remember that number. But there are 20 different amino acids. And so what this means is that there are going to be a number of codons that code for the same amino acid. Okay? For here, since I give you the tRNA, in that table you have, you're actually supposed to use the mRNA. And so what you first need to do is figure out what the mRNA would look like, which is UGG, and then look at the chart to figure out that it's tryptophan that is being made. Okay? 13A, so the attraction between the nitrogenous bases, that's hydrogen bonding. Remember, hydrogen bonding is pretty weak, and therefore that allows the double strands to break apart if we ever need to copy or do anything like that. Okay. For B, it's a peptide bond. That's not new. To make 5 amino acids, remember, to make 1 amino acid, I need 3 nitrogenous bases. So to make 5 amino acids, I need 15. Okay, this is the central dogma. The whole point of this entire chapter is that we're going from DNA to RNA and then into proteins. And finally, frameshift mutation, I already went over it, but it's basically messing up those 3, the codons that you look at. So after a frameshift mutation, all of the amino acids are messed up after that point. Okay. For 14, remember, karyotype, we're looking at just number of chromosomes, while a pedigree, we're looking at the family history. So if we're talking about autosomal or sex-linked diseases, it's going to be a pedigree. While if we're talking about a chromosomal disorder where you have 1 too few or 1 too many chromosomes, then we look at a karyotype. And so here are the diseases we looked at. These are the 3 that are point mutations. Cystic fibrosis and sickle cell anemia are recessive, while Huntington's is dominant. These are chromosomal because Turner's, you only have 1 X chromosome. Klinefelter's, you have 2 X's and a Y, so that's trisomy. And Down syndrome, you have 3 chromosome 21s. Okay. These are all the different phenotype blood types that you can have. Okay. And then I'm going to go through this pretty nice and quickly with you. So, Jeff and Trish are expecting a child. So Jeff, his father is homozygous type B. So please show the big IB, big IB. Okay. His mother's type O, so there he is. So you know that he has to be IB little I as a result. His paternal his paternal grandmother, okay, so his dad's mom is was homozygous type B, so IB. Trish's father, so Trisha is here. Her father was type A blood, and her mom was type O. Okay, and so we don't know if her dad is big IA, little I, or big IA. We're not sure. Okay, her mother's brother was big IA, big IB, and the reason why this is important, that tells us what her parents are. Her maternal grandfather, right? So her mom's dad was type A. So since she's O, her grandfather has to be big IA, little I. Since this one's IB, he has to be IB, little I. So that helps us there. However, for Trish's father, we're still not sure, and therefore we're not sure what her genotype is because it doesn't actually say. So she could be big IA, little I, or little I, little I. So all the different possibilities for their kids. Their kid could be. Sorry, hold on 1 second. Sorry about that. So their kid could either be big IB, big IA, so AB blood, could be IB, little I, so B blood, or they could be O. Or they could also just be straight A. So there are all those possibilities for their kids. Okay. Number 9. So Essie, Mrs. Essie is type A, so we're not totally sure about her genotype until we keep reading, but we know that Mr. Essie's little I. Since their son, Mark, is type O blood, you therefore know that Mrs. Essie has to be IA, little I, for that to combine and make O blood. You know Luke isn't their kid because there is no B allele here with the parents, so there's no way he could be the kid. Okay, here you've got the mom is O, so little I. The dad is AB, so big I A, big I B. The baby's genotype could be either of these two, so that it doesn't prove that the baby was switched, but it also doesn't prove that they are the parents. Okay, if the baby had been sorry, one sec. Oh, yes, because the baby is B blood. If the baby were O blood, then that would prove that it had been switched at birth. However, since it's B, that doesn't prove it's the baby. So that's the funky thing about this. Okay. Here, if I have O blood, these are the people I can give to. Remember, positive can give to positive, negative can give to positive, but positive cannot give to negative. Okay, so they can receive from O negative or O positive, which is awesome. AB can only give to AB negative and AB positive. They can receive from anyone who's negative. They can't receive from anyone who's positive. So those are the options there. Okay, let me know if you disagree with anything. Just come in. Coagulate means it's going to clump, so it can coagulates with A and RH, which means it's A positive. For sex-linked diseases, the letters don't matter, guys. All that we're saying is when it's sex-linked, make sure to be showing X's, 2 X's for females, an X and a Y for males. Okay. So this is homozygous dominant, so there you go. This would be heterozygous, and this would be recessive for a male. Okay. Here are my answers for the fruit fly. Remember, red is dominant while white is recessive, so those are my answers. Okay. For 16, I've got a red-eyed female and a white-eyed male, so here are all my answers. Here's my Punnett square, and here are my answers. Okay. For hemophilia, the mom's a carrier, so this is her genotype. This is the dad's genotype because he's normal, and here are my results. Okay, so make sure to read the question carefully because this is what's the probability that their son will have hemophilia. So they've got 2 sons. The probability that their son will have it is 50% because it's 1 out of 2. Men with sex-linked disorders give it to their daughters. Remember, they're the ones that give the daughter the second X. Otherwise, it would be giving a Y, and that would be a son. While mothers can give it to either of the 2. Okay. The funky thing about this question, I already gave you the answer. It is recessive, but for you to know, since it's affecting men and women equally, it's going to be autosomal. It's when it affects men more than women that it's going to be sex-linked. Okay, so since it's autosomal, I'm not using X's and Y's. I'm just using these letters. Okay, so look at this answer. Make sure you understand it. This has to be heterozygous. Since this is little a, it's affected. The only one that you're not sure of is this one right here. Okay, because it could be big A, little a, or big A, big A. So show all the options. All right, last one. Woohoo! All right, so here, because if you notice, there are no carriers in men, but there are for women. If there's ever a difference in gender, you know it's sex-linked. And since you have that carrier, you know it has to be recessive. The other thing for you to know, I could have filled this in completely and not shown a carrier. That doesn't mean that it's automatically dominant. I can choose not to show a carrier. It's a pain in the butt. It's mean of me to do, but I can still totally do it, and that's fine. Let's look at this last question. Brown eyes are dominant to blue eyes. Okay, this is a recessive autosomal, so I'm not dealing with X's and Y's. Okay. So you read through it, a brown-eyed woman, so she's brown-eyed, whose father had blue eyes, which tells you she has to be heterozygous, right, because she got a little B from dad. Marries a brown-eyed man whose parents were also brown-eyed. So, okay, the reason why I put big B, little b here rather than big B slash, and you can see I erased it, their son, the son they have, is blue-eyed, which means they both had to be heterozygous. For the dad's parents, you're not sure whether it's big B or big B, little b, you're not sure. Okay, and same with the mom here. So even though it's not sex-linked, you can still show the boxes, don't show the X's. Okay, and that's